Meiosis. The production of offspring by sexual reproduction includes the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. This specialized kind of cell division that reduces the chromosome number by half results in the production of haploid daughter cells. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key features of meiosis are as follows. Meiosis involves two sequential cycles of nuclear and cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA replication. Meiosis 1 is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. Meiosis involves pairing of homologous chromosomes and recombination between non-sister chromatids of homologous chromosomes. 4 haploid cells are formed at the end of meiosis. Meiotic events can be grouped under the following phases. Meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1. Meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2. Meiosis 1, prophase 1. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into the following 5 phases based on chromosomal behavior, that is leptotene, zygotene, pectine, diplotene and dikinesis. During leptotene stage, the chromosomes become gradually visible under the light microscope. The compaction of chromosomes continues throughout the leptotene. This is followed by the second stage of prophase 1 called zygotene. During this stage, chromosomes start pairing together and this process of association is called synapses. Such paired chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate the chromosome synapses is accompanied by the formation of complex structure called synaptonymal complex. The complex formed by a pair of synapsed homologous chromosomes is called a bivalent or tetrad. However, these are more clearly visible at the next stage. The first two stages of prophase 1 are relatively short-lived compared to the next stage, that is pectine. During this stage, the 4 chromatids of each bivalent chromosomes become distinct and clearly appears as tetrads. This stage is characterized by the appearance of recombination nodules, the sites at which crossing over occurs between two non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved is called recombinase. Crossing over leads to the recombination of genetic material on the two chromosomes. Recombination between homologous chromosomes is accomplished by the end of the pectine, leaving the chromosomes linked at the sites of crossing over. The beginning of diplotene is recognized by the dissolution of the synaptonymal complex and the tendency of the recombined homologous chromosomes of the bivalents to separate from each other except at the sites of crossovers. These X-shaped structures called chiasmata in oocytes of some vertebrates, diplotene can last for months or years. Final stage of meiotic prophase 1 is dikinesis. This is marked by terminalization of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of dikinesis, the nucleolus disappears and the nuclear envelope also breaks down. Dikinesis represents transition to metaphase. Metaphase 1. The bivalent chromosomes align on the equatorial plate. The microtubules from the opposite poles of the spindle attach to the kinetochore of homologous chromosome. Anaphase 1. The homologous chromosomes separate while sister chromatids remain associated at their centrosomes. Telophase 1. The nuclear membrane and nucleolus repair. Cytokinesis follows, and this is called dyad of cells. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between two meiotic divisions is called interkinesis and is generally short-lived. There is no replication of DNA during direct interkinesis. Interkinesis is followed by prophase 1, a much simpler prophase than prophase 1. Meiosis 2. Prophase 2. Meiosis 2 is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast to meiosis 1, meiosis 2 resembles a normal mitosis. The nuclear membrane disappears by the end of prophase 2. Chromosomes again become compact. Metaphase 2. At this stage, the chromosome align at the equator and the microtubules from opposite poles of the spindle get attached to the kinetochores of sister chromatids. Anaphase 2. It begins with the simultaneous separating of the centromere of each chromosome, which was holding the sister chromatids together, allowing them to move toward opposite poles of the cell by shortening the microtubules attached to kinetochores. Telophase 2. Meiosis ends with telophase 2, in which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, that is, 4 haploid daughter cells. Significance of meiosis. Meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved across generation in sexually reproducing organisms, even though the process per se paradoxically resulting in reduction of chromosome number by half. It also increases the genetic variability in the population of chromosomes from one generation to the next. Variations are very important for the process of evolution. Thank you.